Every cell in the brain has a job. Neurons send the electrical signals that shape our thoughts. Glia support a healthy brain environment and protect those neurons from injury. But where do all of these cells come from? Inside the developing brain, neural stem cells act like biological factories. They divide and transform into the many different cell types that make up the nervous system. In the Flanagan Lab at UC Irvine, researchers are studying how these stem cells decide what to become, and how those decisions might help us repair the brain. First, the scientists grow neural stem cells in carefully controlled culture conditions. Over time, the cells multiply and expand, creating enough material for experiments. Researchers then examine the cells to see which proteins they produce, tiny molecular markers that reveal whether a cell is becoming a neuron, an astrocyte, or something else. Understanding these differences is especially important for diseases like glioblastoma, an aggressive brain tumor that often resists chemotherapy. To study these subtle molecular differences, the team combines biology and engineering. They build tiny devices called microfluidic chips. These chips are made from a soft polymer called PDMS that is molded and bonded to a glass surface to create microscopic channels. Once assembled, the device is attached to an electronic chip and examined under a microscope. Now, the experiment begins. Cells are introduced into the device and flow through the microchannels one by one. Because cells carry electrical charge, they move in response to small electric fields inside the chip. Cells with different surface proteins behave slightly differently, changing how strongly they move through the channels. By observing where the cells travel, researchers can separate, reculture, and expand the numbers and analyze the different populations of neural stem cells and their descendants, or drug-resistant glioblastoma. These measurements help scientists understand how brain cells develop, how tumors like glioblastoma evolve, and how the brain might someday be repaired. By combining cell biology, engineering, and advanced imaging, the Flanagan Lab is uncovering the hidden rules that guide how brain cells are made. These discoveries could one day help scientists rebuild damaged brain tissue, one cell at a time.